Okay, so here we're going to both patch and streak some yeast cells. So I've got some sterile toothpicks here that have been sterilized in an autoclave, and a receptacle to catch my used ones. I recycle these, re-autoclave them, and reuse them. And I've got some plates that both have yeast strains on that I want to patch, and some plates that I want to patch too that are pre-labeled here with the particular strain names that I'm going to patch to. And I've got a couple different media types that I'm going to do this to. So I'm going to start with the first strains. I'm going to carefully take a sterile toothpick here, and I'm going to touch, in this case, a single colony from my strain. This one's name is 1398. So I'm going to go and find a nice sort of isolated single colony. It's kind of hopefully away from the rest. Touch that colony. I've got that on the end of my toothpick. That strain is 1398. I'm going to come over here. I like to put my finger where I'm transferring to when I have a bunch of strains on one plate, so I don't lose my place. Flip this over, and I'm just going to make a little line. So I call that a patch when I do that. And I'm going to do that same thing on two other plate types for this particular application that I'm doing. So again, I like to always put my finger where I'm going to when I have more than one strain. Flip it over, make another little patch. And in this particular case, I've got one more plate, the same application. A little patch there. So on two of these plates, that one's backwards, I am going to streak them. So I'm going to come over to that little patch that I made, which may be a little hard to see now. There it is. Where my finger is. And I'm going to take this clean toothpick and make kind of a straight line through that. And now trying to maximize the amount of surface area that that toothpick is touching to the surface of the agar containing plates. I'm just going to kind of smear that out. The goal here is just that I'm trying to thin out the number of cells that are on there. In this case, I might even flip the toothpick over to the clean side, transfer a little bit of those further, and then streak those out further. Turn this upside down so I can make a little more space. And so the goal is of that is to let those cells grow, and then hopefully you can see here's where the cells were put on more densely, and then as the cells get thinned out, you should hopefully have single colonies grow. Those single colonies will arise from a single yeast cell that divide then exponentially to form one of these colonies.